am Sage Steele, ESPN SportsCenter anchor and a new proud board member with the V Foundation. I'd like to welcome you to the 22nd annual V Foundation Answer for Cancer Symposium. During this unprecedented time of cancer and COVID-19, we have virtually brought together some of our most brilliant doctors and researchers and our brightest and most promising V scholars to discuss their latest research. They will also talk about the effects of a pandemic that has caused unparalleled disruption throughout the cancer research community, shuttering many labs and slowing down cancer clinical trial operations. The scientific community must ensure that this pause is only temporary because research and clinical trials are the only way to make progress in developing new therapies for cancer. Given the long timeline between basic cancer research and changes to cancer care, the effects of pausing research today may lead to slowdowns in cancer progress for many years to come. When the V Foundation was formed in 1993, it was Jim Valvano's dream to fund cutting-edge research that would eventually eradicate this insidious disease. Approximately 1/3, that's over 38% of men and women, will be diagnosed with cancer at some point during their lifetimes. So, joining us today are two of the founding members of the V Foundation, former CEO of the V Foundation, Nick Valvano, and the chairman of the V Foundation and former CEO of ESPN, Steve Bornstein. Glad to have both of my friends here. Nick, thank you so much. It is wonderful to see you again.
Thank you, Sage, and thank you for joining us for our virtual Answer to Cancer Symposium. We've been having these symposiums for 22 years, and it's become an integral part of the wine celebration. I remember the first year when we had one. We had 4 doctors, 2 cameramen, myself, and 4 people looking for coffee. We sure have come a long way since then. It's become a very integral part of the V Foundation. Actually, it's what we do. It's why we exist, and we fund innovative cancer research. Hopefully, someday we will find a cure that will save someone you love. Steve, tell us about the initial motivation that led you to the meeting with Jim and Pam.
So in the early 90s, in an 18-month period, I lost my mother, my brother, and my father all to cancer. My mother was from leukemia, my brother was colon cancer, and my father was pancreatic cancer. Around that time, one of the announcers that worked for me at ESPN, Jim Valvano, was diagnosed with cancer, and we came up with an idea that maybe we should start a foundation to raise money to beat cancer and to defeat it. And Jim was very receptive to it, and with the auspices of ESPN, we started the V Foundation in 1993.
You know, Steve, it seems like yesterday when we had our first board meeting in your conference room in New York City. We've accomplished so much since then, and we've awarded $250 million for cancer research, funding the most innovative cancer research at distinguished cancer centers. I know because I used to visit them all. Not that I knew what they were saying, but they motivated me. And every time I left, I became convinced that these men and women were going to find a cure for cancer. Steve, tell us about your idea behind starting the V Foundation.
My idea was we were going to try to raise money to beat cancer or to help suppress cancer. And Jim was very clear that he wanted to defeat cancer. And so we made it, our policy at the time is that we were going to raise money for research, all forms of cancer, and to really try to address how the impact of this deadly disease on so many people touches everybody's lives. Is there a chance for us to cure it in our lifetime?
That's incredible, Steve. Now I'd like to introduce Dr. Bill Nelson, who is the chairman of our scientific advisory board. He has accomplished many things. He's a swimmer, he plays guitar, he sings, he probably dances. I don't know about that. Besides, he's an MD, a PhD, he takes care of cancer patients, and he does research. On the side, he runs the Johns Hopkins Cancer Center. Welcome, Bill.
Well, thanks, Nick and Steve. And I love the V Foundation, and for lots of reasons. First, there's the great science we fund. Second, the sports. Yes, I'm a fan. I love to kick soccer balls. At this point, I have to watch. And yes, of course, I love the yearly wine celebration. And I can't tell you how much I wish I could be in Napa with all of you this year. But we can still raise a glass and unite against the fight against cancer virtually. My experiences in Napa over the years have taught me that All of you know a lot about sports and a lot about the wine, but you've also learned a lot about the science too. And this morning, we'd like to add a little bit to that knowledge. Some of what we'll talk about is the scientific updates that I know you're expecting because you've really appreciated them through the years. But the important message I'd like to get through is just how important it is to attract and keep young investigators in the field of cancer research. These are the best and brightest people who could be making their careers and fortunes in many ways. Thank heavens they've chosen cancer research. And when we fund them as V Scholars, we encourage them to stay for the long haul because we know that they are the key to the ultimate victory over cancer. So how do we find these V Scholars? They're nominated by each of the cancer centers across the country as the best young scientific prospects. And in a competitive review process conducted by our scientific advisory committee, a small fraction, the elite of the elite, are selected to be V Scholars, receiving $200,000 a year for 2 years to kickstart their own research programs and to give them a leg up in securing funds from other sources, truly leveraging the V Foundation investment. In the past, this critical support has allowed V Scholars to think more boldly how to understand, detect, prevent, treat, and cure cancer. But now, in the midst of this COVID-19 pandemic, with many cancer research laboratories across the country temporarily shuttered, clinical trials interrupted, many of us worry if progress against cancer itself hasn't been put on hold. I'm certain we're going to work our way through the coronavirus threat and any future coronavirus surges. What we want is to create with these V Scholars a surge in discoveries, treatments, and cures as the nation's cancer research enterprise roars back to life.
Yes, thank you so much, Dr. Nelson. That information was invaluable, especially to be able to hear it firsthand from you, from a leader at the COVID-19 Resource Center at Johns Hopkins. So thank you for everything you continue to do. Now I would like to introduce Dr. Joe Moore, Professor of Medicine, and Dr. Chris Wood, Associate Professor at Duke Cancer Institute, to tell one V Scholar story. Listen to this.
Thank you, Sage. It's a pleasure to be here, and it's a great pleasure to say that I've been in the cancer research and cancer treatment business since 1975. And in 1993, it was my pleasure to be Jim Valvano's doctor. Since that time, the V Foundation has done a great deal of work in pushing forward research in having many young investigators become better investigators and be funded. And also that has led to improvement in cancer therapy and in the cures for cancer. What I'd like to do is bring two of those things together and introduce you to a friend, a great teacher, a great researcher, and also a person I know very well, and that's Dr. Chris Wood. I met Chris in, I believe, March of 2013, when he had been at Duke for only a short time, and he developed a type of malignant lymphoma in his chest. It's called a mediastinal diffuse large B-cell lymphoma, and that is a potentially curable type of lymphoma. Chris came to see me. He was treated with a very aggressive course of chemotherapy called dose-adjusted or EPOC for 6 cycles. And then after that, he received local radiation to that area in the mediastinum. He has been treated. After treatment, he has been cured of his disease. He is now 7+ years out and is doing extraordinarily well. And amidst all of that, he became a V Scholar, which I thought was a great icing on the cake. And I want to introduce him and have him tell what he does and how that has meshed with how we treated and how he did. He did extraordinarily well, continues to do so. Chris?
Okay, well, thank you very much, Dr. Moore, and it's a pleasure to be here with you today. As you said, I was a 2013 V Scholar, and I'd like maybe for a moment just to take you back to that time. So in 2013, I had just started my independent lab at Duke University as a professor after completing something like 14 years of advanced training in engineering and cancer biology. And I was finally ready to start my own independent program where I was going to have the chance to take on some of the big new ideas in cancer research that I was excited about, to take those ideas forward. I was excited about that opportunity, but I was also nervous. And one of the things that made me nervous at that time was the question of whether funding agencies would be willing to support a young investigator who had some new and kind of different ideas and who came from a little bit of a different background. Um, so around that same time, as you just alluded to, something very surprising and frightening happened to me, and that's that a few months after I started my laboratory at Duke University as a faculty member, I received a really frightening cancer diagnosis. And one of the silver linings of that experience, something that makes me smile when I talk about it, despite the fact that it was a scary time, is that through that experience, I had the good fortune to meet you, Dr. Moore, and be cared for by you, and to ultimately to achieve a cure because of my care through that you provided to me. One of the stories that I think a lot of people enjoy hearing about my experience as a V scholar, but also as a cancer patient, is one that I remember fondly, and that involved a visit that I made to your clinic for a regular checkup. This was around the time when I was actually taking intensive chemotherapy, and I'll never forget it. I came into your clinic, and I had had scans done, and you came into the office to see me, and you said that you actually had two pieces of good news for me. The first was that my scans looked great, and I was responding well to therapy. And the second was that I had been awarded a V Scholar Award, which was the first grant to support my lab's research at Duke. That award ended up having a huge impact on my career. First of all, it funded breast cancer research in my laboratory that led ultimately to discoveries that are now forming the basis of a human clinical trial. But more generally, that funding catalyzed our ability to explore several additional new avenues of research in the lab that led to really a number of other exciting discoveries concerning things like how cancers grow and regulate their growth and death processes, how cancers overcome the effects of chemotherapy treatments, and how we can leverage this understanding that comes from these studies to design new types of therapies. And today, I'm extremely gratified to say that there are now multiple clinical trials going on around the world to advance some of the discoveries that we made that were originally supported by the V Foundation. And there are also multiple biotechnology companies that have been founded to create medicines that are built around these advances. And so, for me, the V Foundation and the V Scholar Award program have had just enormous positive impacts on me, the same way that you have had an enormously positive impact on me, both personally and professionally. And my hope is that I can continue to pay these things forward in the form of continuing to advance cancer research.
Thank you, Joe. And thank you, Chris, so much for telling us your amazing story. This is really a testament as to why funding young investigators is crucial now more than ever before. On that note, I would like to introduce you to Dr. Shruti Nayak, assistant professor at NYU Langone, an inspiring female scientist who is truly opening doors for women in stem cell research. Dr. Nayak, please give us a quick overview of your research.
Thank you so much for asking, Sage. So the work we do is really about the future of cancer and how we can prevent cancer. So I'm an immunologist, which means I study the immune system. And one of the things that we're trying to understand is right now we're trying to fight cancer with the immune system, but there are still properties of the immune system that can promote cancer formation. So can we understand what those properties are that cause cancer to form and in fact stop it in its tracks even before it forms? And so that's sort of the dream for the future, is how do we stop cancer before it even becomes cancer?
Absolutely. Dr. Nayak, I'd like to ask you though, what is it about the V Scholar Award that is so important to you personally?
So I am an assistant professor, as you introduced me, and this is a really early phase of my career. And what the V Scholar Award really allows me to do is ask big, bold questions and take risks and go outside the box to solve one of the hardest puzzles of human biology, that's cancer. And so this is really, really important funding at this stage of my career to take those big risks and ask those big questions.
And that brings up something that's very important to me and to the V Scholar program, and that's about funding young cancer researchers, especially women, especially people of color. Why is that so critical to discoveries?
So I think diversity is very, very important when we talk about discovery. Because, as I mentioned, cancer is one of the most complex biological problems that we have faced. And we want as diverse of a group of people trying to solve this problem to get to the answer as fast as possible. So, this means bringing in people from very different backgrounds with very different training, and bringing in and keeping women and people of color and other underrepresented minorities because they bring a unique perspective, a unique value, and unique solutions to this extremely difficult problem.
So there's nothing like hearing from these brilliant young people themselves, is there? I'd like to introduce you to some more V scholars and let each of them take a minute to give you a sense of just what the great brain trust is that we're building. We've asked each of them to give answers to these questions. What fundamental scientific question are you chasing after? Second, if everything works as planned, what impact will it have for cancer patients in the future? Third, give us a sense for how getting a V Scholar Award was helpful to your career. Why is it important to support V Scholars? And finally, why is it so important to fund these early career investigators now in the time of the COVID-19 pandemic?
You've probably heard that almost every patient who gets diagnosed with pancreatic cancer will die of their disease. But I bet that you haven't heard that we already have a treatment that can cure some patients without surgery or long hospital stays. This treatment is called stereotactic body radiation therapy, or SBRT. It sounds really fancy because, well, it is. SBRT is an ultra high tech approach to aim high energy X rays at tumors anywhere in the body to completely kill them. The catch with pancreatic tumors is that they sometimes grow very close to the intestines, which are very sensitive to radiation. Because we haven't had a way to protect the intestines from this side effect of SBRT, we haven't been able to bring this important advance to patients.
So, Dr. Taniguchi, if everything works as planned, then what impact might it have for patients in the future?
So when I explained this problem to the V Foundation in 2015, they gave me a grant to find an answer to this problem. And we searched into the greatest depths of cells to understand how we could kill the tumors with SBRT without damaging the nearby healthy bowels. And we eventually found an answer. We could use a medicine already in the clinic to rewire the oxygen sensing proteins in the cell. And when we did this, it puts up a protective shield that reduces damages from X-rays. And the best part is that this only worked in healthy cells and not in tumors. So this made our SBRT more effective and actually helped us cure some patients with this disease. These successes were made possible by the V Foundation. They allowed our lab to flourish and get grants from the NIH and other resources. We also work with drug companies to bring this medicine to patients, and we've started 2 clinical trials already, and we'll hopefully have the answer to whether this might lead to more lives saved from pancreatic cancer.
So there is a silver lining there. That is wonderful news. Thank you for sharing that with us. And I'd like to ask you how the V Scholar Award helped you specifically with your career.
Early funding from the V Foundation had a compounding effect on my career since I spent less time writing grants and more time doing innovative science, which helped our technology flourish.
Timing, of course, is everything. And I'd like to ask you why it's so important to fund these early career investigators right now during this crazy time with the COVID-19 pandemic.
The massive financial losses caused by the pandemic has stopped cancer research in its tracks like nothing else in history. Universities, hospitals, and even some philanthropic organizations have been very squeezed financially. And this has ended some science funding and some jobs for brilliant, brilliant people. You've heard of government bailouts for large corporations and small businesses, but there's almost nothing planned to help cancer science rebuild. When the funding stops, so does our progress against cancer. If you don't support foundations like the V Foundation, it's not crazy to think that cancer research may be permanently harmed. The V Foundation may be at its most important juncture in its history. they can continue to fund innovative scientists like me, who would have otherwise had to stop their research or possibly even lose their job. It's possible that a donation now to the V Foundation may help preserve and support an idea that could save thousands of lives in the future.
And that is our main goal, saving lives. COVID-19 has had a profound impact on everyone, especially cancer research centers and their investigators. So now I'd like to introduce Dr. Jose Trevino. He's a 2015 V Scholar from the University of Florida. And Dr. Trevino, what is the overarching question that your research is trying to answer right now?
And so the overarching theme outside of pancreatic cancer or within pancreatic cancer is cancer health disparities. And that's been the main focus of my research, is looking at differences amongst different races, ethnicities, and determining outcomes. And from that, trying to decide whether or not there is a biological component that might allow survival in certain patients and outcomes, poorer outcomes in others. Unfortunately, Black patients or African American patients here in the US have the worst outcome with pancreatic cancer, followed by whites. And then, interestingly enough, Latinos do the best. And therefore, a big focus of our research is to try to figure out what differences we have amongst races. God made us all very special. And therefore, when we look at the differences amongst each other, amongst the races and ethnicities, we might be able to determine certain targets or certain rationale for why certain patients do better than others, whether it's socioeconomic, whether it's biologic, or whether it's other factors that we aren't really paying attention to in the research, I think is an important question to ask. And I think that doing this research and determining these outcomes and determining the answers to these questions will determine the outcomes and hopefully be able to increase our survival rates amongst all patients with pancreatic cancer.
Absolutely. And pancreatic cancer, we all know how devastating that is. Thank you so much, Dr. Trevino. Now let's hear from Dr. Gianna Hammer, a 2016 V Scholar from Duke University. Dr. Hammer, what is the fundamental cancer question that your research is trying to answer right now?
I like to draw an analogy with sports because in sports, there's a playbook and an attack plan and a strategy to win. And in the battle against cancer, it's the immune system that makes up this attack plan. And so in order for the immune system to win, it really has to understand what kind of cancer it's up against and design the right place. So what we want to answer is, how does the immune system battle against colorectal cancer? Which immune cells are really designing the playbook? And which plays need to be strengthened in order to win the battle?
All right, so what do future seasons look like? What is the hope that patients will gain from the research that you're doing?
Yeah, the hope. This is really core. This is really core to anyone who pursues cancer research in the way that I do. So understanding who writes the playbook and what are those plays, that gives us new things to work with in terms of building new therapies, attacking cancer in different ways, making new and maybe even unexpected strides towards reaching a cure.
Dr. Hammer, as heavy as all of this is, I know that there is also some excitement involved when you're making progress. What can you share with us?
I think one of the most exciting and perhaps unexpected discoveries that we've made so far is realizing that cancers in the gut, such as colorectal cancer, really abide by a different set of rules as you might think are already set by cancers of the skin, for example. So the relationships between one immune cell and another is just so unique in the intestine. We wouldn't have predicted that certain cells are connected to one another and dependent on one another. These relationships we found only exist in colon tumors. And through this discovery, I think we have the fundamental understanding of deciphering how to manipulate these relationships in ways that are helpful to how the immune response wants to really attack colon tumors.
Dr. Hammer, we appreciate you so much. Our strategy to win is to keep great researchers and investigators like you in the game. So don't go anywhere. Next, I'd like to introduce Dr. Bolkin Felis, a 2017 V Scholar from the Jackson Lab Cancer Center in Bar Harbor, Maine. Dr. Bolkin Felis, what is the fundamental cancer question that your research is currently trying to answer?
The goal of my research is to understand how cancer treatment can cause side effects and long-term health problems in young cancer survivors. And in particular, I'm interested to know how these cancer treatments that are designed to save life can damage reproductive system and lead to infertility or premature menopause in cancer survivors.
As always, this is all about hope. So, what can you tell us? What is the hope that patients will gain from the research that you're doing?
So if we succeed understanding how all this happens, we may be able to develop strategies and preventive treatment to protect reproductive health, to protect ovarian function, and give the chance to have own families for young survivors that underwent those damaging treatments.
Dr. Bolkin-Felis, it's about funding and it's about timing, and especially timing right now in this COVID-19 pandemic. So can you talk about the importance of funding these early career investigators now?
I hope what we've learned from the COVID pandemic is also that there is a need to fund a larger variety of research. So we are ready for the next health crisis. That could be the next pandemic or low fertility rates in our aging population. So I think this reflects that we cannot just really focus on one research area, but be ready to have basic understanding from all potential diseases. So what I'm trying to tell my trainees, what excites me, is that our research may lead to actually helping patients to have healthy families. Often we hear that cancer survivors suffer in their later life from various side effects. And one of them that is not often discussed is fertility loss. So we can imagine a young cancer survivor in her 20s that is trying to start her family, but she finds it difficult. And maybe at the time when she was undergoing cancer treatment, nobody told her that her reproductive abilities may be destroyed. And nobody told her that she could prevent that maybe by freezing her eggs or potentially other methods. So when I'm telling my trainees that all that we do, all this hard work, one day can turn out to a treatment that will help women like that to have their own biological children.
I think we all know women who have suffered unable to have their own children. So this is truly wonderful news, and our prayers are that that continues to come to light. Now you're going to hear from our final two researchers, 2018 V Scholar, Dr. Daniel Lee from UVA Health System, and Dr. Jamie Spangler from the Sidney Kimmel Comprehensive Cancer Center at Johns Hopkins. Dr. Lee.
Great, thank you. Well, and thanks for the opportunity to discuss what we've been doing in the lab with you guys today. I heard growing up, "Kick it up a notch" by the infamous chef Emeril Lagasse, who makes wonderful food. But I took that to heart, and when I got into my fellowship in pediatric oncology, I knew we had to kick it up a notch in order to cure childhood cancer. So in fact, my project I did is something called CAR T cells, which you may have heard about before. But what we did is we took the immune cells from a patient and we kicked those T cells up a bunch of notches. And in fact, we made what I think is the most powerful immunotherapy that's known to man right now. I did this for childhood leukemia before coming to UVA. And in fact, we treated the second kid in the world with a CAR T cell for leukemia. And I just saw the amazing potential that this kind of therapy had to cure really refractory and highly relapsed cancers. And so here you can see that this is a brain scan of one of the kids that I treated. And he had one of the worst leukemias I've ever seen. And it was all throughout his brain, all this bright whiteness that you see on the surface of the brain, those squiggly lines, and then inside the brain. That's all disease. And you can see that even though we gave him some of these CAR cells in his vein, they trafficked to the site of his disease in his brain and eradicated the tumor there. That was amazing. So I thought, well, if we can do that, then why can't we do that for pediatric brain tumors? Why can't we do them for pediatric brain tumors outright? That's about the time I came to UVA. And since I've been here, And with the help of the V Foundation, we've designed, engineered, and manufactured a car for the worst childhood brain tumor ever called DIPG. Every kid who's diagnosed with DIPG dies within 9 months of their initial diagnosis. That is awful. And nothing works for this disease. But our new car cures mice that have DIPG. And oh, by the way, they also cure glioblastoma, which you may have heard about. That's a brain tumor that's really bad in adults. So it works for pediatric and adult brain tumors. And we're even still looking to see if we can treat other kinds of diseases with this kind of therapy. So now we patented it, and we're going to use UVA cell production facility to take this discovery directly to the patients. I want to start a first-in-human clinical trial of these CAR T cells for kids with DIPG at the University of Virginia. It could be the first treatment ever to actually help these kids, to hopefully cure these kids. And how awesome would that be? Because without you, these kids wouldn't have a chance.
Absolutely incredible. And to know that we are getting that much closer, that is priceless. That is everything. How about you, Dr. Spangler? I know that you're pushing as well. What fundamental scientific question are you chasing after right now?
So I'm very interested in engineering natural proteins to do useful things as potential drugs or therapies that could activate human immune cells. And the project that I'm working on right now involves using what's called a cytokine, which is basically a protein that floats around your body and activates different immune cells that activate a response against cancer or any sort of invading pathogen. And the problem with the natural version is that it can activate both cells that will attack the pathogen as well as cells that will protect the pathogen. And what my research is trying to do is to specifically activate only those cells that will attack and kill the cancer, so that we can specifically target disease and lead to a therapeutic response in patients.
So if everything works as planned, and of course that is always the hope, Dr. Spangler, if it goes as planned, what impact might it have for patients in the future?
So the beauty of this type of research is it's very general. So it's not targeted toward just one type of cancer or even just a few types of cancer. It could reawaken the immune system against literally any type of cancer that would potentially be able to then respond and generate an effective destructive response against the cancer. And so the idea would be that this would be a technology and a mechanism that we could use and broadly deploy in a variety of different patient settings, and even amongst patients who may have different types of cancer within a particular cancer type, right? So there could be patients who have breast cancer, right? But not all breast cancers are the same. And the idea is that our technology could be the ultimate equalizer in that it would be effective not only across different cancer types, but across different patients with the same cancer. And it would be also the type of therapy that would not be something that you would need to have expensive manufacturing or deployment, which can be a limitation for a lot of therapies. This would actually be a natural molecule that's already in your body that we've made a few tweaks to. And now we can actually deliver that as literally an oral medication potentially that you could take as a drug that will cure the disease.
Dr. Spangler, thank you so much. That is such exciting news. And we know that you will continue to push for just that, a cure. You know, every year in Napa, one of the best parts of the Answer to Cancer Symposium is asking our esteemed scientific advisory board members to respond to your questions about cancer. Since we aren't actually in the room with you today, we have asked in advance, though, for some questions and have posed them to the smartest doctors in the world, V Foundation scientific advisors, to give you those well-deserved answers. And this question is about breast cancer. So I've asked Dr. Nancy Davidson from the Fred Hutchinson Cancer Research Center to respond, because Dr. Davidson, we've traditionally only heard about one type, one form of breast cancer, but it sounds like now there's so many more types. Take us through that. How many are there?
For a long time, we did think there was only one kind of breast cancer, that the cancer started in the breast tissue and then developed from that. But now we know there are really many types of breast cancer. And that's so important to us because it reflects the biological origins of these different types of breast cancers. But more importantly, it helps us to talk with women and men about what the natural history of the breast cancer might be and what kinds of therapies might be the most appropriate for that kind of molecular type of breast cancer.
What exactly does that mean?
For a long time, the pathologists could tell us that breast cancers look like they start at either the ducts or in the lobules of the breast. But with time, we came to realize that there were biological qualities of these breast cancers that we could measure. For example, some breast cancers make proteins called the estrogen and progesterone receptor proteins. This is very important to us because it says that those breast cancers might be responding to the estrogen that the woman's or man's body makes, and that we should then use anti-hormone therapies or endocrine therapies to try to treat this kind of breast cancer. Other breast cancers make the HER2 protein, a different kind of growth stimulating protein. And we know now that those breast cancers are best treated with anti-HER2 types of therapies. And then finally, some breast cancers don't make any of those proteins. And they might be the ones that are the best suited right now to treatment with chemotherapy. So understanding these subtypes of breast cancers is extremely important to us because it helps us to tell patients about what their prognosis might be. And very importantly, it helps us to help them select the kinds of therapies that they should take. Now, this is getting ever more sophisticated. So we're going beyond one gene or two gene or one protein or two proteins to sometimes molecular tests that look at multiple parameters simultaneously. For example, the Oncotype test or the other types of molecular tests that can help us to tell patients about the best types of therapies for them. It's a very exciting time because we're developing a more precise way of counseling individual patients.
Yeah, it is an exciting time for the advances in research and that patients, that they have options and that the therapies can be customized to them specifically. So what is the first question that patients want to ask you though? What is it?
The first thing that patients always want to know, of course, is what their prognosis is. What's likely to happen with this breast cancer? And that's extremely important. But we also want to talk with them about prediction. What might be the best kind of therapy? So traditionally, we have felt that the hormone receptor positive breast cancers might be the best breast cancers to have, in the sense that they often have a very long natural history, and they're often helped by hormone therapies. I've been in the breast cancer field long enough that I know the time when we thought that HER2 positive breast cancers had a terrible outlook. But now with anti-HER2 therapies, we have much better therapy for these types of breast cancers and therefore much better outcomes. Where we still have some lack right now is with the triple negative breast cancers. Frequently, they are responsive to some kinds of chemotherapies, but sometimes they're not. And so we're excited that other therapies are coming on board right now that might be appropriate for some of these triple negative breast cancers. For example, in some cases, if the tumor has a BRCA mutation, a BRCA mutation, we might think about the PARP inhibitors. And that's also the kind of breast cancer where the new immunotherapies, the checkpoint inhibitors, seem to have the most impact right now. So knowing these subtypes is really important because it helps you to think about what might be likely to happen with the breast cancer. But more importantly, what's the best therapy that we can use to try to make the best outcome for that patient?
And are breast cancers similar to other kinds of cancer, or is it in a league of its own?
That's a really interesting question. We've always thought about cancers as coming from the cell of origin, the tissue from which they derive. For example, breast cancers come from the breast, prostate cancers come from the prostate, colon cancers arise in the colon. One thing we've come to realize, though, is that sometimes the molecular pathways that have gone awry in cancers can be the same in different kinds of cancers. For example, we now know that the BRCA1 and BRCA2 mutations that we frequently see in breast cancer, or sometimes see in breast cancer, are also seen sometimes in ovarian cancer, and they're seen in prostate cancer. And the BRCA2 mutations can also be seen in pancreatic cancer. So that's extremely important to us. It's vital information because it suggests to us that therapies that are directed towards these BRCA pathways might actually be useful across different cancer types. So this is what we call tissue agnostic types of therapies. I think we're going to see more of this. For example, stomach cancer can sometimes have the same HER2 abnormalities that we see in breast cancer. And so some people with stomach cancer who have those abnormalities should take the same kind of anti-HER2 therapy that we might normally use in breast cancer. So it's a really exciting time because we're looking at the intersection between anatomy, the tissue of origin, and biology, the pathways that are involved, and trying to make them integrate. But today, when a patient comes in with HER2 positive breast cancer, I'm able to tell her that we have an enormous medicine chest of anti-HER2 positive therapies that will be available to her. That for early breast cancer, we'll be using them right from the beginning to try to reduce the possibility that the breast cancer would ever return. And if perchance she's in the minority of women where the breast cancer does return, we have a large number of agents and drugs that are now available to help that patient turn that metastatic cancer into a more chronic treatment. So even in the short time of my career, a few decades, we've seen how great science has translated into medicine that's making a difference in people's lives today.
All right, thank you. So from breast cancer, we received many questions about childhood cancer. And I've pulled some of these questions together to ask Dr. Mike Kasten, who runs the Duke Cancer Institute, to give us some answers. And it is clear, Dr. Kasten, that much progress has been made in pediatric cancers over the last 3 decades. But there's several questions I have for you here. What's left to be done? Can children's cancers be treated like adult cancers? And why do we need special tests? I know there's a lot of questions, but that's the thing with childhood cancers. We need answers.
So those are great questions, Sage. People usually think of cancer as a disease of adults. And it is true that the incidence of cancer increases with age, especially rising precipitously after the age of 50. But there are more than 11,000 children in the United States who get cancer each year. And that number is just the ones under the age of 15. If we count older teenagers, it's even a larger number. However, it's important to understand that children get very different types of cancers. And that's shown on this first slide, that pediatric patients get different types of cancers than adult patients, with adult patients mainly getting what are called carcinomas of the lung, the breast, the colon, prostate, and so on. But children, the most common tumor that they get are leukemias and lymphomas. They also get a relatively high incidence of brain tumors. And then they have tumors that adults don't get, things like neuroblastoma and retinoblastoma and Wilms tumor and certain types of rhabdomyosarcomas or tumors of the muscles. So they are very different types of tumors in children versus adults. But in addition, there's actually a cancer peak that occurs in early childhood. And that's shown on this next slide, with the majority of children that get cancer having them between the ages of 2 and 6, because that's a time when they're going through very rapid development, and the kinds of cancers they get are a result of abnormal development of those tissues. Historically, a diagnosis of cancer in a child was virtually a death sentence. In the early 1960s, the survival rate for a child diagnosed with cancer was about 4%. That meant that over 95% of children who were diagnosed with a cancer would die. But new successes in treating pediatric cancers in the 1960s and 1970s really were instrumental in driving the revolution that became the war on cancer. Using combinations of chemotherapies and radiation therapy and surgery, all combined in carefully designed clinical trials, drove the cure rates for pediatric cancers as a whole from approximately 4% in 1960 to about 80% today. However, cure rates have largely plateaued in the last 20 years, with particularly disappointing lack of improvement in patients with high-risk malignancies, especially those that metastasize to other sites. And so there's a lot that we have left to do. In addition, although we may now cure a majority of children with cancer, that is not without risk. Current therapies have significant side effects, often leading to life-altering disabilities as children grow into adults. And this includes significantly increasing chances of developing a second or third cancer as they get older. Also, survivors of childhood cancer can have significant problems with many of their organs, their heart, their lungs, their kidneys, and they have a high risk of developing diabetes down the road. So in order to change this paradigm, we desperately need new classes of therapeutics so that we can not only get back to a trajectory of improving survival of children with cancer, but also decrease the physical, emotional, and financial costs associated with the current treatment regimens that rely on standard chemotherapy, radiation therapy, and surgery. So I'd like to show a series of cartoons to sort of illustrate where we were and where we're going. And in this first cartoon, one of my favorites from Gary Larson, of cavemen looking through their hands at rocks, and he calls them early microbiologists. That illustrates where we were in 1960 with cancer. We did not have the information to even know what questions to ask about cancer. And even if we knew the questions, we didn't have the technology to answer them. But despite that, as we developed some drugs, we were able to combine them in clinical trials and have some successes in the 1960s and 1970s. And that's sort of illustrated in this next painting from N.C. Wyeth, where it's a man who's blindfolded trying to find his way through. That's the way we figured out how to use various chemotherapies and radiation therapy and surgery to cure a number of tumors. So through very rigorously designed clinical trials, we figured out how to use these approaches, although there were toxicities. And that's illustrated in this next slide of the killer macrophage, that treatments would unfortunately not only kill tumor cells, they would also kill the normal cells. So it's amazing that despite these non-selective treatments, that because we figured out how to combine these various agents, we did have amazing successes. Again, because of the combination of therapies and testing them in clinical trials. But since the 1970s and 80s, with support of the National Institutes of Health, and since the 1990s with the support of V Foundation and others, the biomedical community has learned a tremendous amount in the past 30 years about the differences between normal cells and cancer cells. And that's illustrated in this last slide that shows how treatments have progressed from radiation therapy to chemotherapy to now targeted therapies and immunotherapies that are much more selective for treating the tumor without and sparing the normal cells. So we began to develop these ways to kill the cancer cells more specifically, sparing damage to the normal cells. And this is particularly important in children who are still going through their growth phases. They're still developing, and they will live for decades after their treatments. So reducing the toxicity of treatments is even more important in children than it is in adults. In 2020, on average, over 80% of children diagnosed with a cancer will survive their disease. Our goal, with the help of the V Foundation, is to bring that up to 100%. And at the same time, make the therapies more specific so that only the tumor cells are killed by therapies, resulting in less short-term and fewer long-term toxicities. We want our children to be cured of their cancer and to go on to live happy and healthy lives for decades to come.
And Dr. Kasten, because of you, I think we all truly have hope that we will someday make that happen and take care of these kids. Thank you for all that you're doing. No symposium is complete without discussing immunotherapy, and it seems like it's a recent phenomenon, but it's really not. So we are asking our resident expert, Dr. Olya Finn from the University of Pittsburgh School of Medicine, to fill us in. And Olya, many people are asking why everyone with cancer doesn't just get immunotherapy. So can you give us that big picture and then talk about CAR-T as a specific kind of immunotherapy?
So it seems like a recent phenomenon, but it isn't. You're so right about that, and exactly the point I wanted to make. When our most prestigious scientific journal, Science, put immunotherapy on its cover page in December 2013 as the breakthrough of the year, my reaction was exactly that. Maybe breakthrough of 50 years, if not more, of cancer immunologists trying to show that the immune system is there to also protect us from cancer, not only from viruses and bacteria. And in fact, here's the timeline, and it starts in 1890s. When I was starting my career, like our V scholars that we just heard from, there was no V Foundation and nobody to encourage young people to go into cancer immunotherapy. We were being told by our older colleagues and well-wishers to change our interest, or we would be committing a career suicide. Well, fortunately, they were wrong, and many of us persisted, like Jim Allison and Tatsuo Honjo, who received the Nobel Prize in 2018, and sent a message loud and clear: we can beat cancer by strengthening the immune system. So immunotherapy is now a first-line therapy for many cancers, and either alone or in combination with standard therapy, it extends life and provides cures. Patients can be given drugs to help them generate their own antibodies and T cells against cancer, or can be given cancer-fighting antibodies. You all heard about Herceptin, for example, for breast cancer. And T cells can be given that are engineered to see their cancers, so-called CAR T cells. CAR, by the way, stands for chimeric antigen receptor. And you ask, why doesn't everyone get immunotherapy? Well, if you ask me, I would say everyone should. And definitely before any other therapy that might damage the immune system beyond what cancer has already done. But we are learning that some patients and some cancers do not respond to immunotherapy. And that's important to know for two reasons. Patients who are not responders and that to immunotherapy should be promptly treated by some other drugs that could help them in order not to lose precious time. But the other reason is cost. Immunotherapy is very expensive. There are many people here and around the world who could be helped by immunotherapy, but individuals as well as whole countries cannot afford it. So how do we solve this problem? How do we deal with that? One way is to hope that as more immunotherapy drugs become available, and they're constantly being developed, and a lot of our scholars, for example, are working on new ways to provide immunotherapy, the price will come down. But the other way is to try to prevent cancer. A vaccine would be a much cheaper way to deal with this long-lasting cancer epidemic. In fact, cancer pandemic, there isn't a corner in the world that is not affected by cancer. So we're all following coronavirus vaccine development. 150,000 people in this country have died already from this virus, and we all want to stop the carnage. But cancer kills 600,000 people in the US every year, year after year after year. The cancer pandemic has been with us too long now. So once we get that COVID-19 vaccine, which we will, this preventative vaccine, we should become just as interested in determined and resolve to go after a cancer prevention vaccine with the same urgency, because we know how to cure some people, and that's a big achievement. But we can try to eliminate cancer completely, and that will be an even bigger achievement. And as a cancer immunologist, I believe that cancer immunologists can give that gift to humanity. And if you're wondering how close that might be, it's much closer than it appears.
See, optimism. We are so thankful for that. Thank you, Dr. Finn. And now I would like to ask a global leader in medicine and preventative oncology, Professor Funmi Olapati. Dr. O, thank you for being here. What do we know, first of all, about cancers that run in families? And then what about the change in our lifestyles, things that we can do to maybe prevent cancer or at least catch it early on?
Thank you for asking. I've spent my entire career as an oncologist looking for the root causes of breast cancer and why cancer runs in families. And more than 2 decades ago, we found the BRCA1 gene, and then BRCA2, and now many more genes that we're characterizing that predispose individuals to breast cancer, ovarian cancer, colon cancer. In fact, the hottest area of research now is trying to figure out how we can intercept cancer in families where we know people are born with a predisposition. The good thing is not everyone who's born with an inherited risk factor for cancer ends up developing cancer. And that's why I'm really excited to join the research to find new ways to prevent cancer in all high-risk populations. My work has taken me all the way back to Nigeria, where I came from originally, across to India, to China, to Brazil, all over the world. We're looking for ways to understand how cancer develops so that we can figure out how to prevent it in all populations. And your generous donations to the foundation makes it even ever more closer for us to find ways to prevent cancer.
We could not be more grateful to the incredible work done by our Scientific Advisory Committee. Nothing shows the value of our V-Scholar Awards like someone who was honored with this award and used their V-Scholar Award to start a remarkable career in cancer research. Dr. Lisa Cousins is a tremendous scientist whose research home is the Oregon Health and Sciences University. She earned her V-Scholar grant in 2000, and many projects, teams, discoveries, awards, and innovations later, she became an esteemed colleague on the V Foundation Scientific Advisory Board. Lisa, tell us about what the V-Scholar grant meant to you, what you've been working on since, and where you see cancer research going in the future.
Okay, hi, Bill. Really good to see you today. Thank you for asking me to talk about the impact of the V-Scholar Award on the research out of my laboratory. I think, as you know, the V-Scholar Award was my very first grant as an independent PI a long time ago. And the grant was written to study proteases and proteolytic mechanisms in solid tumors. And the importance of that grant was that it provided my young lab with a great deal of flexibility. Whereas we thought we were going to study proteases, what we very quickly discovered was that the proteases we were interested in were secreted by immune cells, normal inflammatory cells that surrounded and were interdigitated in solid tumors. And the grant, because it afforded us flexibility, let us turn our attention to why were there immune cells within the solid tumors. And as you know, my lab has focused on chronic inflammation and the role of the immune cells. And what we have discovered over the last 20 years is that those immune cells that are early in cancer actually help cancers grow. And without the V-Scholar grant, that let us pivot a little bit to look at inflammation instead of just the proteases. We've spent almost 20 years understanding how we can target immune cells in cancer and what kind of biology those immune cells promote to help generate solid tumors. But also then when we're successful with treating cancer patients with various kinds of therapies, we can now use those immune cell biomarkers that we discovered so long ago to actually understand if a patient is responding to a therapy or perhaps has a cancer that's resistant to that therapy. And that then lets us enroll those patients onto different drug regimens that will hopefully be more successful in helping them to live with or eradicate their tumors.
So the question is not if we will see a victory over cancer. The question is when. And when we do, what will that look like?
I absolutely think we will see victory over cancer, and I hope we see that in my lifetime as a scientist. The revolutions in the last 5 years with just targeted immune therapy has completely changed the name of the game, in that we now have long-term survivors of patients that were initially diagnosed with malignant metastatic melanoma. Used to be just a 6 month to 1 year survival outcomes. These people are now living, when they respond to these immune therapies, they're living 5 and 10 years out. So that is just frankly remarkable. But what we need to be able to do is expand that out to all different types of tumors. And where the victory will be is understanding personalized medicine so we can treat each patient's tumor individually so that we know how or who will respond to what therapies, how we can leverage in targeted immune therapy and enable people to live with cancer as opposed to die with cancer.
Dr. Cousins, I have to say, as someone whose father is battling cancer, living with cancer, that is something that is music to our ears. So thank you for all that you do there. I mean, with everything going on though right now in the world currently, the COVID-19 pandemic, how has that affected cancer research, and why, in your mind, is it important now, maybe more so than ever before, to support cancer research?
Yeah, the last 4 months of research in North America, but internationally, has really ground to a halt, except for those laboratories that were doing active COVID-19 related research to identify targets for vaccine development. For most of us cancer researchers, the biggest impact has been, well, first, an inability to be in our labs doing research. And the ability to maintain momentum. As a chair, in addition to having my own laboratory and overseeing my own trainees, I'm responsible for an entire faculty of junior scientists, people that have just started their own labs. My career, I can survive this. I've established my relationships. I have my camaraderies. My lab will be fine. But for the junior trainees that have now been outside of their labs for 4 months, away from their trainees, away from their colleagues, I am very worried about their ability to sustain their careers. I think in my career, there's never been a more important time for partnership with philanthropic organizations and partnership with people that can be generous with their philanthropy to help us make sure we don't lose an entire generation of really talented, motivated, and passionate young researchers that simply can't make it through this tremendous hiatus and hurdle that COVID has put on all of us.
I love this wine celebration event, even if it's virtual. Next year, I hope to be there and give you each a hug. But for now, did we answer your questions?
Well, Bill, you did good. You didn't answer them all, but we'll get some of them next year. But what on a serious note I'd like to say is over these years, I've never gained more respect for all of our scientific advisory board and all the scientists for what they do. You know, I'm often asked, what would Jim say? How the hell I know? I don't think Jim knew what he was saying half the time. But I will tell you this. I know he would be happy. I know he'd be grateful. And after thinking about it, he'd probably say, what's taking you guys so long?
You know, I never had the pleasure of meeting Jim, although from what I've seen, that doesn't sound like you're too far off there, Nick. Here's the thing, and I think we all agree after listening to all of this tonight, I have hope that it's not going to take us too much longer. This has been such an enlightening event. Thank you to the doctors for taking the time to educate us on the advances of cancer research and inspiring us to keep the momentum pushing forward, moving forward every single day. Now it is time for our audience to weigh in. And I think that is so important because the audience always has questions for our doctors. And if you're thinking of them, please drop them in the comments below. Meantime, let's take a look at some of the questions that we have received so far.
If Jim Valvano were diagnosed with his cancer today, how would his treatment go? And would he have had a positive outcome based on the advances in research and treatment since 1993?
Sure. No, I think that's a question that I've been asked many times. If Jim was diagnosed today, would he do better? I think unequivocally the chance of him getting better, even an outside chance that he would be cured, would be much more likely now, based on many of the discoveries in terms of the biology of cancer, some of the things that you've done, many other researchers have done, that have brought other treatments to light and to the clinic that our menu of things to treat with and to have a better outcome would be much better now than it was then. And Jim always said, why isn't there more? And my answer to that, if he were here today, he would say that that's great, you've done a great job, but it's not enough.
Does having a history of cancer increase your risk for complications from COVID-19?
With the advent of COVID in a time of cancer treatment, we've had to think a lot about whether or not having a history of cancer increases your risk for acquiring COVID or having a poor outcome with COVID. I think that's a work that's very much in progress right now. Some of the early information that came out of China suggested that individuals who had certainly certain types of cancers like lung cancer might be more likely to have poor outcomes with COVID. As we see more about what's going on in the United States, I think that the jury, in my mind, is still out. We know that a number of organizations, including the National Cancer Institute, ASCO, private groups, the COVID and Cancer Consortium, are trying to gather that information as we speak to be able to help to counsel people who are cancer survivors about their risk of COVID. I think the important issue here, though, is that all of us are at risk for COVID. And so every single one of us should be taking all of the precautions with regard to hand hygiene, with regard to physical distancing, with regard to all the safety issues that have been recommended for all of us. So nobody is immune to COVID.
If a person is about to start cancer therapy, should they consider postponing treatment due to COVID-19? What if the cancer patient has been infected with COVID-19?
So I think in these cases, hospitals have taken a lot of precautions and have very stringent guidelines for whether you should go ahead and get therapy for your cancer treatment or be cautious of your COVID and get COVID therapy under control. When there was so I'm from New York, when there was a peak of COVID in New York City, elective surgeries were put on hold because the risk to the patient is so high of getting COVID. So I really suggest that you go with your hospital guidelines and your doctor's guidelines because they have your best interest in mind. And they also understand the data and the dangers that are current to your area, your location, and in terms of COVID cases that are currently ongoing and your specific therapeutic needs.
Does having received chemotherapy or radiation in the past raise your risk of getting COVID-19 or having a more serious course of the illness?
All cancer patients are at an increased risk of side effects from COVID-19. If you've had chemotherapy or radiation in the past, you could be at increased risk of bad side effects from COVID and possibly even death. But it is largely dependent upon how recently you've had your chemotherapy or radiation. As you know, those treatments can suppress your immune responses and may make it more difficult to fight off the virus. So I would highly recommend that you speak with your oncologist and local primary care doctors to get more specific answers to your specific situation.
How has COVID-19 affected clinical trials in your area?
Because this is a very highly contagious virus, because COVID-19 is a very highly contagious virus, clinical trials were put on pause at many institutions, particularly in New York, where there was a huge peak in COVID cases. And this was really to protect patients from exposure to sort of this viral infection. And now as New York numbers are coming down and we've really flattened the curve, we're wearing masks, and following sort of public health guidelines, those trials are coming back online because the risk to the patients is much, much lower, and people are sort of following public health mandates. So that's how our city and our institutions in this city have responded to COVID-19 to best protect cancer patients.
I know this is very far-reaching, but when are we going to get a cure for cancer?
We already do have a cure for cancer. What we have is a cure for a cancer, because cancer is not one disease. Cancer is many diseases, and so So we are slowly marching through various different cancers and trying to get the cure. And then I always say, of course, but wouldn't it be nice if we didn't have to deal with that incredible variety, but prevent them all to begin with?
What are the causes of cancer in both children and adults?
Virtually every cancer is caused by a combination of your genetics and your exposures. And in children, the genetics is more important. In adults, probably the exposures are more important, such as smoking or high-fat diet or other things that contribute to certain types of adult cancers. In children, it's always hard to pinpoint. And it's clear that genetics plays a more important role in the development of childhood cancers.
With the world we live in now with COVID-19 pandemic, why now more than ever is it important to continue to keep funding young cancer researchers?
So COVID-19, oh my gosh, what a shock was that. You know, we were turning away in the lab, producing a lot of data and moving along very quickly, and then all of a sudden COVID hit. And unfortunately, like everybody else across the country, we had to shut down. That meant our experiments stopped. You know, and we lost 3 months, at least at this point with that. But we're losing a lot more than that because it takes time. Even we can get back in the lab tomorrow, it takes time to rebuild the progress that we were at, to get back on track. So 3 months, a delay is really more like 6 months delay. So you can pretty much double almost any time out of the lab because it takes just that much more time to get the experiments back up and running. But the clock still ticks, right? Kids are still getting cancer, but the discoveries have stopped cold turkey. So we need to invest even more now than we did before in order to try to catch up, to make up for that extra lost time and to get those discoveries back on track.
Well, COVID has us all shut down or shut off temporarily. But as you know, people are battling against cancer still today and tomorrow. And Jimmy V and the V Foundation tell us to never give up. And nobody understands this more than early career scientists such as myself. We want to succeed. We want to make a mark. We want to continue. We want to do something that's important. And that's what is, I think, so important for funding early career scientists at this time.
What can we do to prevent cancer?
Yeah, so when I first started my career, I was hunting for the gene that predisposed to melanoma, only to discover that even if you were born with a gene that predisposed to melanoma, if you didn't spend a lot of time in the sun, you were not likely to get melanoma. So it's really important for us to know that there's some modifiable lifestyle risk factors that we have identified. We know that excessive alcohol increases your risk for cancer. And we know that eating too much, and we all do sometimes, can make you gain excessive weight. And obesity is a very important risk factor for cancer. So in my work, I've really emphasized prevention because I was taught in medical school that prevention is better than cure. So let's all go out there, have fun, eat and drink in moderation, and then avoid excessive sun exposure. All those things really help us reduce the risk of cancer. And by the way, did I say tobacco? Smoking is a very important risk factor for cancer, and we can all do better by not picking up that cigarette.
When looking at cancers, can we reduce cancers to a set of common traits?
That's a great question. I think that reducing cancers to some common traits is incredibly important, right? And when you look at patients who have a certain subset of cancers, and if you can extend that into other cancers, the therapeutics development can be amazing. I mean, you develop one drug and you're able to tackle 3, 4, 5 different types of cancers. And so I think that's an incredible opportunity. The reality is, though, in my opinion, is that it's a little bit more complicated than that. as I've matured more as a researcher and as an oncologist, I recognize that outcomes are different amongst not only different cancers, but different patients. And so the complexity gets further and further. But we need to understand that complexity to really be able to develop the therapeutics that are necessary. And so as I train my residents and my students and even the younger faculty, is, in my opinion, for example, pancreatic cancer within itself has a subset of different types of traits, different types of characteristics that will make some more vulnerable to therapies than others. And I think that knowing that, and I think that getting deeper and deeper into that allows us to understand this.
What about probiotics in our diet? Can they help us fight cancer?
Of course, I don't have these answers, but I do get that question, and it's reasonable because the intestine is home to trillions and trillions of bacteria. So what I will say is that there's really no way to separate diet and its impact on the bugs and bacteria that live inside of us. And while there may not be one true cure that you can gobble down and cure anything, it certainly can't hurt to think about supporting communities in your body that are linked to doing good things.
Can you cover why immunotherapy has become the hot button when people are talking about cancer treatment?
So the thing about cancer and what makes it so pernicious is that it's your body's own natural cells going haywire, right? Essentially getting out of control. And so the idea that we can actually use the body's natural immune response, right? Something that we already have inside of us, and activate that to control and counter the disease just brings a whole new perspective onto the cancer therapeutic landscape. Rather than trying to fight against cancer and fight against this non-natural enemy, we can actually use something that we already naturally have built in to go to war against this unregulated version of our natural cells.
What are the most exciting new therapeutic developments in cancer research today?
One of the answers that I often give to people is reflective of my own belief, and that is that there isn't one answer. There are certainly there are so many different therapeutic modalities that are being developed right now that are showing promising activity in different contexts, and I think ultimately what this speaks to is the idea that the fight against cancer and the curing cancer is going to be require a multi-pronged effort. And what's really exciting is that we're making such extraordinary advances really across the gamut, from diagnosis to biology to therapy, whether it's direct therapy, targeted therapies or immune therapies or other types of modalities. And I think that's something that I always try to convey to people. If there's something to be excited about, it's that we're not just relying, I think, on one type of advance. It's that we're making advances on so many different fronts.
Chris, I think you hit on something else that's very important in terms of our ability to diagnose. We always called Jim's cancer unknown primary. We did not know where it came from for sure. We had ideas, but this was prior to PET scans, prior to some of the things that we can do now, pathologically to identify, and by identifying better, to be better able to craft treatment and to craft treatment that would specifically be targeting his particular cancer. And many of those things just simply weren't available at that time, but due to research, much of that has come around since then. And I think we would be better both in terms of diagnostics, but also therapy as well.
Mm-hmm. And that brings up an interesting point that that I mentioned. So, I started doing cancer research in the early 2000s as a graduate student. And I can tell you that the papers that were written in the early 2000s, not that long ago, almost feel like the Stone Age compared to what we understand now. The pace of advances in our understanding of cancer biology and genetics is just mind-blowing. And so I think that's, it takes time for those scientific advances to be translated into clinical advances. But they certainly are a positive sign for the future.
Couldn't agree with you more.
This is definitely a question for you, Nick. Think back in time to when Jim was diagnosed, when you sat with Pam, you sat with Steve, and you conceived of the V Foundation. Did you ever think it would become something like it is today?
To be quite honest, when we met as a family after Steve approached Pam and Jim, I'm not so sure we had any aspirations to do anything but do something for somebody we love. And what happened is the number of friends, associates, family members, corporations that took up Jim's fight was awesome. And the number of people who have volunteered every year for 27 years, giving up their time and obviously their money and their talents. I don't think we could have envisioned that. Secondly, we didn't know what the hell we were doing. We were just crying. That first meeting in Steve's office, the first hour, we were just everybody telling stories about Jim and sobbing, and because we were all friends. Everyone was asked by Jim to be part of that. I can remember that meeting like it was just the other day. So I would like to say, after all the years, the pain's gone away, but it hasn't. It really hasn't. And things like this make it hard for me because it makes me, reminds me of what we're missing. But the V Foundation would not be anywhere without our board of directors, the scientific advisory board, and all the people who hold fundraising things in their driveway to come out and shell a lot of money to drink wine in Napa Valley. I mean, that's the range of our supporters. Pretty much reflects who Jim was. He had this range of people and supporters and friends, all levels of society. So for me, this is just awesome and unexpected. The only bad thing is he should be here for it.
Nick, trust me, you are not the only one getting choked up, getting emotional. I will say this, though. I happen to believe that Jim is watching all this from above, all the hard work, and also watching the fact that you cry in supermarkets still and probably having a nice chuckle out of it, deservedly so. Thank you. And thanks all of our guests and our audience for joining us for this momentous event. We have a really exciting lineup of entertainment to continue with the Wine Celebrations 2020 virtual event. Next up, the V Foundation Wine Celebration and ESPN. They've teamed up for their first ever V for Victory telecast with my friends Kevin Agandi and Hannah Storm. That's at 2 o'clock Eastern Time, 11 a.m. Pacific on ABC. This telecast will shine a light on cancer survivors, their stories, and the urgency to support cancer research during the COVID-19 pandemic, and of course, to support the V Foundation's mission to achieve victory over cancer. And then we move to our main event, the innovative virtual auction gala. That experience begins at 7 o'clock Eastern Time, 4 o'clock Pacific on the Wine Celebration YouTube channel. Access is available by private link only, so please visit www.winecelebrations.org for further details. Thank you again so much for watching, for listening, and learning right along with us as we all try to eradicate cancer. I'm Sage Steele. We'll see you back on ABC in 30 minutes.